Welcome student to another video of photosynthesis part 3. In this video we will discuss about the dark reaction or the biosynthetic phases which are going to use ATP and NADPH. The product of light reactions are ATP, NADPH and oxygen. In dark reaction ATP and NADPH are utilized to synthesize a food and oxygen is released out. This phase does not directly depend on the light reaction, but it depends on the products of light reaction. It can be verified as follows. Immediately after the light becomes unavailable, the biosynthetic process continues for some time and then stops. If light is available, the synthesis starts again. Carbon dioxide assimilation during photosynthesis occurs through two ways. One is C3 pathway, another one is C4 pathway. The C3 pathway is also known as Calvin cycle because Melvin Calvin discovered this cycle. The first product in C3 pathway is 3-phosphoglyceric acid, which is 3-carbon compound. And in C4, it is 4-carbon compound, which is oxaloacetic acid. That's why the name are been given C3 and C4. Let's start with the C3 pathway, which is also known as Calvin cycle. It occurs in all the photosynthetic plants, that is in C3 as well as in C4 pathways. It has 3 stages: carboxylation, reduction, and regeneration. Carboxylation of RUBP, that is ribulose biphosphate, which is a 5-carbon ketose sugar, is the primary acceptor of carbon dioxide. It is the most crucial step because carbon dioxide fixation takes place here and the 5-carbon sugar is going to convert into 2 molecules of 3-PGA in presence of an enzyme RUBP carboxylase. RUBP has a tendency of oxygenation as well. That's why the enzyme is also called Rubisco. Rubisco is the most abundant enzyme present in this world. The next step is reduction. It is a series of reactions that leads to the formation of glucose. Here, 2 molecules of ATP are utilized for phosphorylation and 2 molecules of NADP are utilized for the reduction of carbon dioxide molecules. Fixation of 6 carbon dioxide molecules and 6 turns of the cycles are needed to remove 1 glucose molecule from the pathway. The third step is regeneration of RUBP. It is important part of the cycle because if 5-carbon RUBP is not available, then fixation of carbon dioxide will not take place. And for regeneration of a RUBP, it requires 1 molecule of ATP. Hence, for every carbon dioxide molecules, 3 ATP and 2 NADPs are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 glucose molecule, it needs 6 turns of this cycle. What does go in and come out during this reaction, during this cycle? 6 molecules of carbon dioxide get in, 18 molecules of ATP are utilized, and 12 molecules of NADPH are utilized in Calvin cycle. And the product formed is 1 molecule of glucose, 18 molecules of ADP, and 12 molecules of NADP. The other pathway is C4 pathway, which is also known as Hatch and Slack pathway. It is present in plants that have adapted to dry tropical regions. They also use C3 pathway as main biosynthetic pathway because large cells around the vascular bundles of C4 are bundle sheath cells. Such anatomy is known as Kranz anatomy. This anatomy where bundle sheath cells are covered by the mesophilic cells. The bundle sheath cells may form several layers around the vascular bundles. They have a large number of chloroplasts thick walled and they are impervious to gas exchange and without any kind of intercellular space. The steps of Hatch-Slack pathway are given below. The primary carbon dioxide acceptor is phosphoenol pyruvic acid, that is phosphoenol pyruvate. It is 3 carbon molecule seen in mesophilic cell. The enzyme for this fixation is PEP carboxylase, that is PEPK. The mesophil leg Rubisco enzyme. The C4 acid, which is oxaloacetic acid, is formed in mesophil. Now, this oxaloacetic acid gets transported into bundle sheath cell, but first it has to convert into either the malic acid or aspartic acid. In the bundle sheath cell, the C4 acid breaks down and it undergoes decarboxylation to release carbon dioxide and C3 acid. The C3 carbon dioxide will undergo in the Calvin cycle and 3 carbon acid will convert into PEP again and it's transported to the mesophilic cell. Now here the bundle sheath cells are rich in Rubisco, but they don't have PEP case. So there won't be any fixation of carbon dioxide in bundle sheath cells. So this PEP is transported into the mesophilic cell where it will again start its fixation, transportation, decarboxylation, transportation and regeneration. The C4 plants are special because they have a special type of leaf anatomy and this anatomy is known as Kranz anatomy. They tolerate higher temperatures. We can see such plants, they can grow at the higher temperatures. They show a response to high light intensity. They lack photorespiration. They have greater productivity of biomass because there is less chance of photorespiration. Now let's learn about the photorespiration. In Kelvin pathway, that is C3 pathway, RUBP combines with carbon dioxide, right? So it gives 3 PGM. Active sites of Rubisco, which is an enzyme, can bind with oxygen as well as carbon dioxide. Rubisco has a greater affinity for carbon dioxide than oxygen, right? So it's always a competitive binding. Now, if in cell concentration of oxygen and carbon dioxide changes, it may lead to photorespiration. Relative concentration of oxygen and carbon dioxide determine which one will be binding to the enzyme. In C3 plants, some oxygen binds to Rubisco because at some point oxygen concentration increases comparing to carbon dioxide. So hence carbon dioxide fixation is decreased. Here RUBP binds with oxygen to form one molecule of phosphoglycerate and phosphoglycolate. This pathway is called photorespiration. In this pathway, there is no synthesis of sugars, ATP and NADP. Hence, the photorespiration is a wasteful process. Rather, it causes the release of carbon dioxide by using ATP. So it's just an opposite, right? So it's useless. But in C4 plants, photorespiration does not occur because they can increase the carbon dioxide in their bundle sheath cell at the enzyme site. This takes place when C4 acid from the mesophil is broken down into the bundle sheet to release carbon dioxide. And this minimizes the oxygenation activity of Rubisco. Due to the lack of photorespiration, the productivity and yield are better in C4 plants comparing to C3 plants. In addition to these plants show tolerance to the higher temperature. So C4 comparing to C3 are better in terms of yield productivity. Let's see the difference between C3 and C4 plants. C3 plants, photosynthesis occurs in the mesophyll cell, whereas in C4 plants, it has mesophilic cell as well as bundle sheath cell. Kranz anatomy is absent in C3 plants, whereas it is present in C4 plants. RUBP is a primary carbon dioxide acceptor in C3, whereas in C4 it is PEP, that is phosphoenol pyruvate. 3-BGA and 3-carbon compound is the first stable product in C3 plants, whereas OAA, that is oxaloacetic acid, is the first stable product. Chloroplasts are only one type. Here it is dimorphic. Photorespiration loss is high because respiration takes place here. Here it is negligible or absent. High carbon dioxide compensation point is required, whereas they need low compensation point because in it itself they release the carbon dioxide and it utilizes it. The optimum temperature for photosynthesis is about 25 degrees Celsius, whereas the optimum for photosynthesis is 35 to 45 degrees Celsius. So these C4 plants can tolerate the high temperature. Photosynthetically, C3 plants are less efficient and productivity is low. Here, C4 are photosynthetically more efficient and productivity is also high. The examples are given of C3 plants are rice, wheat, bean, potato, whereas C4 plant examples are maize, sugarcane, amaranth, and sorghum. So from these difference and comparison, we can conclude that C4 plants are better in terms of productivity and yield. Thanks for watching.